Our next speaker is Dr. Stacey Williams from the Institute for Socio-Ecological Research and Coastal Survey Solutions.
Thank you. Good morning. Today I will be talking about the Diadema restoration project in Puerto Rico that I've been carrying out for the past 4 to 5 years. So as we all know, and the reason why we're in this room today, that we've seen dramatic changes on coral reefs in the Caribbean and in Florida. We're seeing a significant reduction in coral cover, and we're seeing this increase in disturbances like coral disease, bleaching. In Puerto Rico, we're seeing at some reefs really high abundance of fleshy macroalgae, and now we're seeing this passionalid Ramicrusta popping up on all these reefs, which is known to overgrow and kill corals. So the herbivore densities on the reefs in Puerto Rico is relatively low, especially Diadema populations. They still not have recovered from pre-mass mortality densities. Typical reef, you would see less than an individual per square meter. So why haven't Diadema recovered throughout the Caribbean in Puerto Rico? And I tried to answer this question in my dissertation back in 2010. I looked at the larval distribution and also the settlement distribution of Diadema in La Parguera, Puerto Rico. And what I found to be the bottleneck of Diadema in their recovery was the high mortality of settlers and recruits in La Parguera, Puerto Rico. But I also was able to find a hotspot of settlement at a shelf edge location in La Parguera. So I was thinking to myself, how can we increase, kind of reverse this bottleneck or increase the survivorship of settlers and recruits of Diadema? And that's how the Diadema restoration project came about. So the overview of this project is I collect the settlers at this known hotspot. I bring the settlers back into lab. I grow them into young adults to larger sizes, and then I transfer them to the reefs. So the objective is to boost the populations of Diadema around Puerto Rico. And the overall goal is to enhance reef resiliency by decreasing algal cover and also hopefully increasing coral survivorship and coral recruitment. Some advantages of this project is that I bypass a really difficult stage, as Brian mentioned, of rearing the larvae into lab. I'm collecting actually the settlers from the wild and bringing them back. I'm also bypassing possibly this natural predation stage where they're getting picked off when they're smaller by growing them larger in lab and transferring them as larger individuals. Also, I'm introducing a new population. I'm not taking from a healthy population and redistributing them to different coral reefs. So here's a video of me collecting the settlement plates. So I have a couple papers out there that describe the methodology. So as I mentioned to you before, we focus at the shelf edge. That's where we found the hotspot of Diadema. As Brian also, we found that at the shelf edge, there's a low density of adult Diadema. So they are settling, they're just not making it to adults because of the triggerfish there. So we have scaled up this project this year. Before we would have 10 mooring lines, now we have 30 mooring lines. And on each mooring lines, we have 20 settlement plates, and the settlement plates are made of this astroturf. You could buy these doormats at Kmart, and I cut them into smaller pieces. So we tried this year leaving some of the plates out for 2 months because we usually collect every month, and we found that with the 2-month settlement plates, there's a higher amount of algae on these plates and also a higher amount of micropredators like crabs and worms. So it seems to be ideal to leave these plates out for a month. So I collect these plates, as you can see in this video, in dry bags, and these plates are suspended in the water column. What I found was there was a peak settlement at 9 meters in the water column, and I collect during the summertime. That's the peak settlement for Diadema. So far, we've collected more than 2,000 individuals, and we still have a couple more months of collection. So I bring the plates back into the lab. The lab is situated at the University of Puerto Rico in La Parguera, Puerto Rico. This is the outdoor facility. It's nothing fancy. And we collect about 10 lines at each collection, so 200 plates with 4 people. It takes us most of the day. and we go through each individual plates and pick off the settlers. Now the settlers are red, and they're really small. The test diameters can range between 0.4 millimeters to 1 millimeter. When they're at this size, they're really sensitive to everything, sediment, to water changes. So this year we have now focused on increasing their survivorship by placing them in a tank that has a closed system so we can monitor the water parameters. And so far, knock on wood, it has been working for us. So we condition this tank a couple months beforehand to have this nice cover of algal turf so they can eat. And then when the urchins grow to a size that I can visually see them between 5 to 8 millimeters in test diameter, I then take them out to the outdoor facility, which the tanks are on a semi-closed system. So fresh seawater is flowing into the tanks and being filtered through a sock, but yet it's being recycled in case anything happens to the university's pumps. So I feed the urchins. So I feed the urchins twice a week, usually on Mondays and Fridays, and I give them a whole array of algae. Try to help them adapt to what life would be like on the reef. I clean the tanks once every 2 weeks, but when they're relatively small, I clean them every week and make sure that there are no other micropredators like crabs or worms, fireworms in the tanks. So these urchins are in these tanks until they reach a young adult stage. So they grow to 3 to 4 centimeters in test diameter. And this can take up to a year. However, this can be less if you give them enough space and you give them a lot of food. They actually grow really quick compared to, I collect all different species on these settlement plates, especially sea urchins. Echinometra and trypnoces are really slow growers. So Diadema, pretty quick. So I'm going to be talking about our last restocking efforts last summer in 2016. So we transferred the Diadema to a back reef in La Parguera. This is La Parguera right here. The red dot is the reef that we transferred the urchins at. It's called Media Luna. So this is a shallow back reef, depth of 5 meters. And at this reef, back reef, it's mostly comprised of dead Orbicella annularis colonies, as you see in this picture. Before taking any of the urchins out to this reef, what I did an initial survey of the Diadema populations and found their populations to be low. And also they were all adult Diadema. I didn't see any juvenile or young adults on this reef. So the dominant benthic substrate at this reef is turf and fleshy algae. As you can see in this picture, it's comprised of mostly Dictyoa. And there are no large invertivore fishes like triggerfishes. What you will see is like bluehead wrasse, princess parrotfish, and also French grunts. Also, before taking out the Diadema onto this reef, what we did was that we installed corrals. So these are just what we picked pretty isolated Orbicella colonies, and we enclosed them with chicken wire mesh. So it was open to the top, so fish can enter or any other organisms that can make it through this fencing. The approximate radius of these corrals were like 1.2 meters. So I had 2 events of restocking that in June 28th and in September 27th, with a total we transferred to this reef was 343 urchins. I also played around with the densities in each corral. So some corrals had higher densities of Diadema while others did not. And what we found was that the ones that had higher densities of Diadema actually did not try to escape. They kind of stuck in that corral and they stayed there for the time of monitoring. We used these laundry basins to take them out to the reef. So we monitored the survivorship, and not necessarily it's a survivorship, it's like the presence, because I don't really know if they were eaten. I only saw 1 pile of Diadema spines throughout, the monitoring. So what we did was we revisited this reef 1 day, 2 days, 1 week, 2 weeks, 1 month, and 2 months after each restocking event. And we counted the urchins inside and outside the corrals. And if they escaped, if they were not tightly tucked into a crevice, we then would move them back to the corrals, to the nearest corral, and then recount that corral. So here are some of the results. So the majority of the urchins escaped within the first 2 days. For the first restocking, yeah, they're really, you know, you would think with the long spines that they would really have a difficult time, but they move fast and they move far. We actually were able to count, you know, these young adults 30 meters away from the corrals. So if you're doing any type of restocking of Diadema, I would suggest going back the day after and 2 days after, because if you don't do that and you go out a week later, you might miss where they're going. So what we noticed that the Diadema within the corrals, that number stabilized after a week. And survivorship was really pretty high compared to other Diadema restoration efforts. We found that within the first month, we were able to locate 79% of the restocked individuals. After 2 months, 75%. And then after the second restocking, we actually, I could go back to the reef probably today and see 76% of these urchins. We did notice that they exhibited homing behavior, so they kind of stayed in their a perimeter of, you know, that coral head within 2 months. So I was just out there a couple months ago doing coral monitoring, and I mean, they were pretty scattered, but not too far from the coral heads. And they're doing a great job at keeping this reef clean, as in this slide. So we saw changes in benthic composition, and these were evident within the first week. There was less turf and less fleshy macroalgae inside the corral compared to outside the corral. So we saw this kind of transition. They would graze in one area, then they would move along. So within the first month, 50% of that coral head was grazed. And then in the second month, we saw 95% of this coral grazed. Pictures are not, oh. Okay, so this is before. This reef is always turbid, so it's really hard to get a clear picture. You can see that there's algae, and here inside the corral, this is actually a damselfish algal patch, and you can kind of see the border of Diadema grazing to this algal patch. And then these last two pictures are a 1 week after placing them on the reef, and you can see how clean the reef is. So this is a picture before and after, and this is 2 months after placing the Diadema at this coral head. And you can see that they effectively grazed the substrate, and they also have removed the encrusting tunicate Trididium. This is a video. So this is a video that I took, recorded. This is right after I removed the corrals. So this is what you will see on this reef. A lot of Dictyota. So this is outside the corral. And you will notice when I start moving to the coral head with the Diadema on it, you're going to see a stark difference in the algae, how clean the reef is. So here's the coral head. And here are the Diadema. And when there are a few on a coral head, they seem to hide pretty well. When they're in a group, they tend to stay on top of the coral head. I guess they don't have a fear of being eaten. So you can see that the substrate's really clean. As I mentioned to you before, in Puerto Rico, we are now seeing a high abundance of Ramicrusta on reefs, and actually on the east coast of Puerto Rico, we're seeing this as a dominant substrate, and this is known to overgrow and kill corals. So I decided to bring Ramicrusta back into lab and see if the Diadema would eat it, because we noticed in the field that around adult Diadema, there was no really Ramicrusta. So as soon as I put that in the tank, like 30 minutes later, 3 Diadema were on top of it, eating it. And then in 12 days, they pretty much broke it down and they pretty much ate all of it. So Diadema can actually be used to kind of hopefully control Ramicrusta in some reefs. And we're going to do that this summer. We're going to transfer some of the Diadema to these really heavily hit reefs in Puerto Rico, mostly on the East Coast. Also, there's a master student, Manuel Olmeda, and he's going to do some experiments in the field to actually look at what is the ideal density of Diadema that is needed to do restoration, because we don't want them really bio-eroding the reef, but also grazing algae. And he's also going to look at the grazing effects on algal abundance and coral recruits, and also the effect of rugosity on their survivorship. So I would like to thank all these people. This project is funded by NOAA, also Recursos Naturales this summer. And if you have any questions, well, I guess we'll take questions, but here's my contact information. Thank you.